Alright, so this is AP Biology Lab 1. First looking at Part A, which is about diffusion. So first thing you need to do is get a piece of dialysis tubing, which should be sitting in a bin of water overnight or for a period of a few hours to make it pliable. Then you need to tie a knot in one end of that tubing. This will allow you to pour glucose in there without it running out the other end. So you do that, you pour your 15 milliliters of glucose that you measured out, you put it in the tubing, and then you take your glucose indicator strip and you're going to dip that in the bag to see what kind of change we get with that glucose indicator strip. Then you tie the other end off in the bag. You want to leave a little bit of room up here in case we get any kind of expansion or in case we get any kind of diffusing across the membrane. So leave some space there for that. Also notice any colors that are happening inside the bag. Then what you're going to do, you are going to grab your glucose indicator strip. You are going to get a beaker of distilled water. You take your glucose indicator strip, you're going to test the glucose in the water. Note your reaction. Take your dialysis tubing with the glucose and you put that in the 250 milliliters of water, and you add 1 milliliter of the IKI, which is potassium iodide. And then you're going to wait 30 minutes. So here's our stopwatch indicating 30 minutes. You can start that if you want. We could sit and wait for the 30 minutes, or we could just move on. So that's what you do. You wait 30 minutes. Then after the 30 minutes, you're going to remove the dialysis tubing from the bag, from the beaker, and notice any changes in color in either the beaker or in the bag. And you're also going to do test the glucose content in the beaker again to see if any glucose diffused out, and check the glucose content in the bag. You're going to have to cut open the bag or untie the bag to do that. Alright, and that is Part A. Part B, talking about osmosis here. First thing you need to do is you get yourself 6 cups. 1, 2, 3, 4, 5, 6 here up at the top, and you label them water, 0.2 molar, 0.4 molar, 0.6 molar, 0.8 molar, and 1.0 molar. Then, in a similar fashion to Part A, you're going to get 6 pieces of dialysis tubing. You can do this one at a time, or you can do this all 6 at once. If you're working with a couple partners, then you should be able to handle all 6 of them at once. If your dialysis tubing does get too dry, you can dip it back in the water, and that should help you out. So, you get your 6 pieces of dialysis tubing, cut them all right about the same length, tie each end off. In the first one, you're going to put 25 milliliters of water. In the second one, 25 milliliters of 0.2 molar solution. Third one, 25 mils of 0.4 molar solution. Next one, 25 mils of the 0.6 molar solution. The next one, 25 mils of the 0.8 molar solution. And in the last one, 25 mils of the 1.0 molar solution. Then you tie off the tops, just like you did before, again, leaving some room for expansion. So, once you tie yours off, and I'm just going to show you with one of these, but you do the exact same thing to all 6 of these. You are going to take your bag, you're going to blot it with some paper towel. Then you weigh it. You want to get as much water off as possible so that doesn't add to the weight. Then you weigh it, you record your weight, and then you put it in your beaker or your cup here. Then you're going to add some distilled water. You pour it in the cup, and you're using distilled water for all the cups. You're going to fill it up about 3/4 of the way, enough so you're covering the dialysis tubing. Also, try not to pour directly onto the bag. You don't want to interrupt that bag at all. So, after you add the water, you're going to let this sit for 30 minutes. After the 30 minutes, you're going to pull the bag out, blot it with the towel again, and weigh it. Once you weigh it, you record your data, And then again, you're doing that for all 6 bags. That brings us into part C, the water potential. For the water potential here, you need to get a potato and a potato borer. The potato borer can be a little bit sharp at one end, so just be a little bit careful with that. But you take the borer, and what you're going to do is you kind of drive it through the potato, and what you're going to end up with is a piece of kind of a cylinder of potato inside that borer. You can use some kind of tool, sometimes a pencil works, sometimes some forceps that you need to stick through the top to push the potato out. But what you end up with is a piece of potato that looks something like these pieces we have over here. You need 4 pieces. You cut them all down to the same length, which the lab recommends 3 centimeters. And then you get those 4 samples, you put them on the scale, and you weigh them. After you weigh the 4 cylinders, you're going to take 100 milliliters of the solution that you have been assigned. So you may, depending on your lab, you may be doing multiple solutions, maybe just 1 solution. But you take that and you're going to pour it into your cup. So you pour that into the cup, and once it's in the cup, you take your potato pieces, you put those in the cup as well, and you're going to cover those with plastic wrap. And how long are you going to do that for? 24 hours. So you do that for 24 hours. Then the next day, you come back, you take your plastic wrap off, you pull the potatoes out, you blot them with some paper towel, and you weigh them. And you're going to take your results and go to the lab, you're going to graph them, and that's it. That's pretty much the whole lab right there.